Hello, good morning, good afternoon. Welcome to this Synomics webinar. This is the second webinar in our series. Today we'll talk about applying the Synomics platform to advance breeding models in plant and animal space. I'm Peter Kristensen. I'm the CEO of Synomics. I have a background in agriculture, veterinary medicine. Started my career on the operational side with livestock, cattle and pigs. I've worked in pharmaceuticals. I have a background in genetics. And more recently, I've been a partner with Wheatchief, who is a food and agtech investor for the last 6 years. I've been with Synomics for a year and a half. Today, I'm joined by our CTO, Professor Haja Kadarmideen, and I'll allow Haja to join us now and introduce himself. Over to you, Haja.
I'm a CTO and a head of R&D at Synomics. Background, a veterinarian, trained as a veterinarian and practiced it for about a couple of years before starting on a journey as a career geneticist, animal breeder. So that's back in 1992, so it's about 30 years in that discipline. The last 20 years of the 30 years, I've also moved down to bioinformatics and computational systems biology across animal and human biosciences. Just before, my career has been a mix of academia, university jobs to government positions to commercial organizations. Multiple countries, but in like Denmark in the last 12 years, and before moving to Synomics, I was a full tenured professor of quantitative genetics, bioinformatics, and computational systems biology. Looking forward to this seminar.
Great. Thank you, Haja.
Thank you.
So what we'd like to cover between Haja and myself today is a brief introduction to what is Synomics and what indeed is the Insights Platform. We'll talk about the application of that platform into breeding models in plants and animals. We'll share a couple of case studies with you, one from dairy cattle and one from laying hens. We'll talk about some conclusions on the technical and commercial outcomes, and then take a Q&A session. So if you would, please hold your questions to the end of the webinar. I should say that these webinars will also be available on social media and on our website for those of you who'd like to revisit, or for those that indeed have missed the opportunity to join us today. So an introduction of the platform. Synomics seeks to provide actionable and relevant insights that helps partners unlock value from large biological data sets. And how do we do that? We do that by applying a proprietary mathematical framework to these large multi-omic data sets. The outputs, as you can see, can be delivered in different forms, anywhere from individual SNPs all the way to QTNs or the application into breeding models. We will sort of touch on that in more detail as we go through the webinar. As you're aware, biology in itself is complicated and it is indeed hypercombinatorial. If you look at combinations, evaluating a 200,000 SNP panel, as you move up in orders, that quickly becomes a hypercombinatorial problem. At the heart of what Synomics is and is developed to be, it's a proprietary mathematical framework that allows you to examine higher order combinations in a fast and very computationally efficient way. It unlocks combinations that were hiding in data sets that you've already amalgamated. You think about the GWAS approach to analyzing data sets. We're able to mine out novel combinatorial networks that are lost in the traditional way of evaluating these data sets in a very rapid and cost-effective manner. we're able to validate those networks for biological relevance. So how do we do that? As I mentioned, we apply a proprietary mathematical framework that's rapid, scalable, combinatorial, and multi-omic in nature. That's great. Lots of companies try to apply novel technology to the food and ag space, large, complicated data sets. What is slightly different with Synomics is it has a very far-sighted investor, and it's been allowed to build a very large team for the development of the company that is a highly skilled team of hands-on practitioners, bioinformaticians, quantitative geneticists, software engineers, business leadership, deep academic experience. So we are combining best-in-class AI, machine learning, a proprietary mathematical framework with a highly skilled team. And how we think about the platform and the application of the Insights platform is we work hard on developing a proprietary framework around the best-in-class technologies to onboard data, smooth API interfaces to take in best-in-class standardized QC for maximum curation of the data set and value extraction. I talked to you about the processing. Haja will get into a lot more detail in that. We use not only our own mathematical framework, we use best-in-class validation methods to scrutinize the outputs and make sure that they are indeed relevant, the insights that we can call. Post-processing, we're able to use NLP, we're able to use pathway, gene annotation to really interrogate and validate the relevance. But more importantly, we've invested quite heavily into what I call the so what, meaning the translation of it's all very well having a lot of SNPs called into a data set, but if you can't translate it into an existing balanced trait selection, it really is meaningless. So we spend a lot of time, and really since our first webinar, we have been probably a year and a half in stealth, working on some public data sets, some private data sets, to fully understand how we would apply the starting point, which is our patented mathematical engine, building best-in-class Lego bricks around it to make this relevant to the field, and indeed make it relevant to the topic at hand today, which is the application into advanced breeding models. So with that, I'll hand over to Haja and let him take you deeper into the framework and a couple of case studies.
Thank you very much, Peter. Yeah, so with that great introduction and the overview of our Synomics Insights platform in terms of what we offer, what it does, I'm going to walk you through our application of Synomics Insights platform to advancing our breeding models and genetic evaluation systems. So before we get into that, just a quick run through the history of selective breeding, starting with mass selection, all the way through merit, blood, and genomic selection. With mass selection, that's back in the 18th century, it started breed the best of the best based on phenotypic characteristics, followed by herd books and breed societies, etc. Went through a merit index or total merit index system, which is still based on phenotypic and familial information to select best breeding individuals. One of the issues with that approach was that you could not unbiasedly and accurately estimate the fixed effects and genetic effects. I mean, fixed means non-genetic effects and genetic effects. Then come the theory that there's an infinitesimal number of genes in the genome each with small and equal effects of variances contributing to the phenotypic variation. That was a pretty good theory to work on a number of advanced statistical methodologies. Then came in 1980s, the best linear unbiased prediction, which basically changed the way we estimate reading values and fixed effects or non-genetic effects. That is still practiced today in various forms, as we will go through that. So that has changed dramatically the way the animal and crop production, including aquaculture, the genetic change that was introduced through that methodology is quite dramatic. But then this was challenged in 1990s that blood-based selection, the basic fundamental theory that there is an infinitesimal number of genes with small and equal effects was challenged through what you call QTL mapping experiments, where the hypothesis were there are fewer finite loci with large effects on the phenotype or disease risks. And we should be able to capitalize on those big gene effects in what is called marker-resisted selection. And the key hypothesis or the fundamental theory behind that is that the assumption was that there is a putative QTL or QTL which can change the phenotypic differences between individuals, but they cannot be directly genotyped or observed, but you can tag them using genetic markers. At that time, there are a few hundreds to 1,000 microsatellite markers, and the principle was based on linkage within families and partly also linkage to equilibrium in a population. And that was running for 10 to 15 years, quite a lot of investment, and a lot of breeding companies took that on board. But the actual impact of that eventually was not very encouraging, partly because the linkage was within families, and the very fact that it depended on the LD or linkage to equilibrium between an unknown QTL and a known marker was broken down, and it was not persistent. It was during this time that genome sequencing technologies were kicking in, and there came a proposal called genomic selection. And that was based on really challenging the marker-assisted selection. Where do we need to depend on LD? Where do we need to depend on finding causal variants? If you have 10x, 100x more genetic markers in the genome, because the chances are that the marker is in tight LD and linkage with a QTL that we cannot genotype. So then this paradigm shift is in directly estimating genetic marker effects on the phenotypic variation and using the marker effects in the genomic brain. Moving on, that was genomic selection leading to 1.0, and that was the high density SNP assays that was a result of genome sequencing projects where we typically had starting with 10K, 50K, all the way to 1 million SNPs in livestock species. It was still based on blood framework, but the key fundamental difference between the ones before and 1.0 is that given the larger reference population that are both phenotyped and genotyped, given that we can train these populations, estimate the marker effects, test for its validity, then using those predictors, we can go ahead and predict the future performance of young selection candidates that are only genotyped but not phenotyped. What that means is that you can make the genomic selection or selection decision at very early stages of individual's life, therefore drastically cutting down the generation interval. The other advantage of that genomic selection is it simply replaced the pedigree-based relationship used in blood with more accurate genomic relationship, thereby increasing the accuracy of predicted reading values. Then moving on to the whole genome sequencing, it went in and added more data to the high density SNP chip assays, because with whole genome sequencing, you have millions of SNPs or millions of variants, and the chances are that you find the causal variant within the genome sequence data compared to the fixed array data. So if we can directly capture the causal variant within the genome sequence data, we expected that to increase the genomic prediction accuracy. So at least that was the hope. So moving on to see that this slide is basically showing, right, we are making genomic selection 1.0, 2.4, but what are the key driving factors that will progress this genomic selection going forward from 2.0 to further variations of that? And in a nutshell, this is a function of our ability to describe and generate genomic data in terms of density and the accuracy, as well as the ability to process this data and the ability to analyze the data. I will take the first one now. And what you're looking at on the left-hand side, on the graph, it shows the cost of genome sequencing. Human example, but it's okay because other species piggyback on that. So back in 2000, where this human genome sequencing project was set up, it was about $3 billion, and the very, very expensive sequencing cost per individual. And that was because it was really old-fashioned, slow-moving Sanger sequencing, and the cost, it took a long time and extreme amount of cost that was involved. Now you're looking at $1,000 genome, and even if we really look at some of the companies, it's just half of that, and the turnaround time is less than a week. So there is dramatic decline in cost of sequencing by introducing next generation sequencing technologies, and that's about 10 to the power 5 decrease in the cost. While at the same time, we have been also witnessing the 14,000 time increase in computational power. And that together, you might have heard about combining these two graphs in the Moore's law, where the cost of there was a prediction of exponential decline in cost of computing power. When compared to that, the cost of sequencing has gone down in a more precipitous fashion that is actually fully overtaking the cost of computing. So with this, two interesting developments, humongous massive computational power, massive ability to create genome sequence data. How are we doing with analyzing these data sets? In interpreting this data, really capitalizing on the benefits of each of these technologies. In my view, there is no big major leap in the statistical methodology or quantitative genetic modeling that will fully capitalize on these breakthroughs. So there's only a limited gradual improvement. Now I want to talk a little bit about something that has benefited from this massive increase in computational power, and that is artificial intelligence and machine learning, which is a part of an artificial intelligence. It really got spearheaded by the increase in computational power. So I would rather say machine learning is not new. It's been there, but there's a re-emergence of these AI machine learning because we have this massive computational power to deal with big data sets. So you have all these circles interlinked, and it gives certain big names, big data, deep learning, data mining. These are all main characteristics of AI. AI is in everybody's life. It's in our environment. It's the way we interact with society and environment and go about day-to-day life. But what impact does it have on life sciences and health sciences? Yeah, we're starting to see quite a lot of impact on life and medical sciences, be it integration of biological data of different types, including genomic data, integration of farming records or electronic patient records to try and drive the biomarker discovery, drug targets, differentiating cell types, predicting somebody's health risk. It's happening, and it's great because it is efficient, it's fast, and one of the important things about this is that it is real time. You can get things done real time, making decisions. So it's predictive, it supports the decision making, it recognizes the patterns, and it mimics the human brain. And in other words, the core idea behind this is keep learning, repeated learning of the data that's important. So right in the middle is a combinatorial analytics. And why is that? That is coming from our proprietary platform in it. For that, before I introduce that, the main caveat or something that people don't realize is that this is as good as the data is. And it could introduce bias. The bias could be introduced right in the way how you define training, how big is the training set, the training data is, because it's got to learn from the training data to be able to make decisions and predict some outcomes. And if you don't get it right, the bias could be introduced also in the model definition itself. So this, if you're not careful, this could end up in a situation where it works in certain populations, certain background, environmental and genomic background, and it may not work in another situation. So it's very, very important to make sure that this doesn't fall apart in that kind of a training and deep learning. Combinatorial analysis, that's our proprietary platform. It is based on some of the neat mathematical construct, including the way to understand what are we learning, and we can monitor in a very supervised way and apply a statistical and mathematical filter to make sure the input and the output is correctly synchronized, that there is no hidden layers with unknown weights and how you get the outputs. So it's a fantastic combinatorial platform. And as Peter mentioned, we use that on top of or before, after, also looking at feature selections where existing state-of-the-art machine learning AI tools are applied to select features that we will then use in the genomic breeding models. So I've spoken about all these fantastic breakthroughs in genome sequencing technologies, computational technologies, AI, machine learning, but are these improvements enough? I mean, are we seeing the material change in what we wanted to see? Take, for example, genome phenome association study, genomic prediction. A lot of studies indicate that the genomic prediction accuracy is not necessarily huge compared to some of the high density SNP arrays we use. At least we haven't seen that capitalization of the sequence variants of very, very large sequence data. So, and the other thing is the core mathematical, statistical, genetic, quantitative genetic modeling is still linear, and most of the industry is still applying linear unbiased prediction. And really, when you look at it, it's not combinatorial. It is still modeling a single SNP effect, of course, in a construct of relationship metrics, but the actual engine is still additive linear calculations. What about AI machine learning? As I mentioned, the key thing about AI machine learning, it is as good as how you define the training data. And it, by nature, it requires a very large training data to be able to get better prediction models, particularly when it comes to combinations and identifying and mathematically defining what is an epistatic interaction. Then it starts to lose its advantage or the edge over some of the known combinatorial analysis that we put in place. And this, therefore, is still inefficient. So putting it all together, what we're looking at is we are in need of an approach that correctly assimilates these breakthroughs and advantages across a number of technologies and be able to combine, integrate, and predict future performance and breeding values. That's where we come in. So Synomics Advance, to me, it's genomic selection 3.0. We are moving ahead, capitalizing on these advances in cutting edge technologies. I want to differentiate in point by point, what is it that this has that the current methodology, the current best practice of genomic selection doesn't? The first and foremost thing is we apply the state-of-the-art combinatorial genomic selection feature engine, and that is statistically and biologically validated features used in an advanced genomic prediction models. And this can be implemented without disrupting the existing genetic evaluation systems. It will be, just as an example, will be a bunch of models, features, and weights that can be easily, straightforwardly incorporated without much disturbance in how this could be run on, let's say, a daily or weekly basis. It takes over the AI, conventional AI machine learning methods in the context that it can work with small data sets as well as the large data sets. But perhaps the most important and critical point that we need to understand here is that the ability to screen for those, among the millions and millions of variants in the genome, what are those combinations that are actually biologically and statistically meaningful? And to what extent their interactions are additive by additive, epistatic variants, which will then go on to explain the additive components of the genetic variants of the population and improve the prediction methodology. It's plug and play because given the same set of models, small tweaks, we can easily implement that. Efficient, low cost, because it makes no big changes in computational infrastructure either. And last but not the least, we have applied in dozens of POCs with a range of SNP density from a fixed assay chip, assays or arrays to whole genome sequence data up to 60 million. So that's a part two. So hopefully I've given you an overview of the advances in different technologies that are critical for genomic selection going forward. But here, wanted to put in two case studies that we have done, one in dairy cattle, the other one in poultry, to show the benefits of the Synomics advance. So the first one is in dairy cattle population. So this data came from our own farm, Grosvenor Farms in England. It's a data set with about 4,000 animals. Of that, 2,800 were multi-lactation, multi-parity dairy cows of Holstein breed mainly. We had genome sequenced these animals, and we had a cutout of 50K, 100K, 777K equivalent of the Illumina chip out of the sequence data. We wanted to choose the representative trait that represents health, fertility, and production. So the health being represented by lameness and clinical mastitis, and the fertility as conception rate to first insemination, and production was milk fat. So what did we find then is using these technologies, the Synomics Discovery Platform, we found anywhere between 13 to about 120 highly predictive SNPs. Highly predictive in a sense that to what extent, how precise they're able to classify the truly lameness animal as lameness or mastitic cow as mastitis versus those as healthy, 95, 98% of the time. Same thing, highly fertile, lowly fertile, highly productive, lowly productive. So obviously these are really a golden handle on driving the genetic variation. But you also wanted to check up on, do they mean anything biologically to the phenotypes in question? So in this particular case, we were able to map those SNPs in the genes and then look at the annotation, the enrichment of pathways, molecular system, etc., and understand that they are indeed relevant to some of these phenotypes that we looked at. Then to give so what is the result of applying this technology to genetic evaluation? I would say that some of these key metrics that we used as a comparison between the Synomics-driven genetic evaluation system in comparison to the genomic blob or GBlob, also a single stop blob as a baseline method. And this involved 5-fold random cross-validation. And I can also talk about some blinded cross-validation if there's an interest later on, but it's a 5-fold random cross-validation. And we're looking at what is the lift in the prediction accuracy for correctly predicting the phenotype classes, be it production or health and fertility. What is the lift in the reliability of genomically estimated breeding values? What is the reduction of the prediction error variance? What is the increase or decrease in heritability? And that means, are we able to capture extra additive variance that is hidden if you were not taking into account those combinatorial and epistatic interaction variants? What does it do with genetic evaluation, ranking of individuals as best to worst? Does it change? How does the genomic breeding value distribution look like? Is it widespread so that we can distinguish the best versus the worst and make selection decisions? And you see these parameters, pretty good lift compared to what the GBlob could do. And I would say that there are some of the things of relative difference. So it's not actual numbers. When you look at reliability of EBV, you're not talking about 100% reliability. It is a relative difference between two numbers that went from 20% to 100%, just to be clear. So the conception rate appears as an example. So we captured more heritability, and we observe capturing extra additive genetic variation, often more so in low heritable, difficult to manage traits, such as disease and fertility than production traits, which are highly polygenic and continuous. But yeah, you see those lift in the accuracy, reduction in error, great change in reliability, monumental change in reliability and rank correlation. The key point here is that the rank correlation is different. That means the top animal is not the top animal in GBlob, but our reliability of EBVs are better, much, much better. So in that case, the commercial decision of what to select and use as a feature parents would be more advisable and highly recommended to go with a ranking that is more reliable. So that's a fact here. So let me move on to the laying hen study very quickly. So this is the public data, 1,000 laying hens. So the family structure is the 92 sires were represented with 800 dams, 600K the avian chip. So we QC down to about half of it. Multiple egg weight measurements across multiple weeks. We are presenting 28 and 56 week egg weight. So what we found here is about 2,000 plus highly relevant variants that similar to what the dairy cow situation is. And also the functional annotation was pointing directly at the gene that will be a key mechanism involved in egg development, growth, etc. Similar to heritability, similar to dairy cow situation, we got a better lift in most important parameters like reliability and prediction error variance. So I think this is also a 5-fold random cross validation. We are looking at the mean accuracy differences. And again, there's a relative change between two numbers. Heritability is about the same, but you do understand that the rank correlation is different and the reliability is higher with Synomics advanced technology. So hopefully with this, I've been able to give an overview of different technological developments and genomic selection 3.0 and what we witnessed in some of the commercial and public data sets. Over to you, Peter.
Thank you, Haja. So what I'll try to do is just briefly conclude and sort of frame the hopefully follow-on discussion from some of the participants on the webinar. What we're focused on, Haja is taking you through some of the technical advancements, and we think very much as this is an evolutionary process, right? We are driving novel tools wrapped around a mathematical framework, best in class, AI and machine learning, highest quality QC and data curation to drive new material insights from large data sets. We are submitting this as fundamentally next generation data analytics, but that is meaningless unless you can drive it to commercial outcomes. So as I mentioned in the beginning, we have very thoughtfully invested into a large team, a team that understands how to work with leading practitioners in the plant space and the animal space to translate this to delta G. Because that is the foundation of the relevance of this as an evolutionary step. Unless you can translate it into existing plant and animal programs to materialized delta G, it does not matter. Speaking as a man who's spent part of his life in a genetics company. So we're very focused on this. And I think it's important to emphasize that we're very thoughtfully trying to be a discovery platform to unlock more reliability, more accuracy, more variance in data sets in animal and plant breeding. A discovery platform trying to unlock new traits, resilience to disease. We will seek to do that all through partnering. We are not trying to be a plant breeding company. We're not trying to be an animal breeding company. We're trying to partner and submit a next gen platform to integrate and to be additive to existing established processes in breeding companies. So we're very much doing this through a partnership approach. We're actively looking for commercial data sets of size to engage with partners in, and we have a fully fledged team. We have people on site in Oxford in the UK. We have people on site in Copenhagen. We have people on site in Madison, Wisconsin. So we are open for business, so to speak. We've tested our platform now for almost 2 years. We have a much better understanding of what it does. And this is really an exciting moment for us because we wanted to engage with the wider community to say, look, here it is. And we're excited to engage in further dialogue. So with that, I will stop talking and hopefully we can take some questions from the participants. Who wants to go first?
Quiet silence.
First one here. Would this replace genomic markers or enhance genomic markers? And that's a Haja question for sure.
I can't see the question. Can you repeat that, Peter?
Would this replace genomic markers or enhance genomic markers?
It would enhance the genetic markers and genomic selection technology.
Next question is, how do you validate the prediction models of these genotypic markers?
Yeah, so we have multiple validation strategy. One, when we are really looking at the discovery side of the platform, and that is even before we integrate that or translate that to genomic prediction models, we have a number of inbuilt prediction validation mechanisms. One example would be the Insights platform itself has multiple statistical methods that checks for overfitting, controlling false positives, increasing power of detecting variants. So there's multiple methods are already built within the Synomics Insights platform. Then we have the results of combinatorial SNPs, for instance. Recorders are highly predictive because they're able to predict the cases versus controls correctly most often and highly accurately and so forth. This is further validated using regularization methods such as lasso or ridge or elastic net, which is sort of in between, to try and make sure that we are not overfitting the models, which often happens when you deal with big data. It's very, very important that these are not ghost variants that are actually correctly predicting or classifying the phenotype or disease in questions. So once we have done that, then we have the highly predictive marker sets, genetic markers, which is then utilized in a genomic evaluation system, or pretty much very similar to blood framework, J-blood framework. There, as you know, this would go through, again, a 5-fold cross-validation, splitting the data sets, randomly finding 5 equal parts, where each data point is used as a test and training set. So we developed those things to look at the level of prediction accuracy, how is the correlation between predicted phenotype and the estimated breeding values. And there are so many number of other things. And the final test, I think that is very, very important to point out, is that we have put through this engine in the genetic evaluation, predicting the future performance of individuals without actually knowing the phenotype through a blinded cross-validation system. In other words, given the genomic data, we can predict the future performance or the phenotype data and actually cross-check with the real phenotype that is observed as a part of the genomic selection procedure. So that's how we built in the validation, statistical validation, and evaluate the prediction accuracy.
Thanks, Haja. Then next one is, does the size of the data set change the signal you're able to find?
We would say that it does. More data points we have both on genomic side, genomic sequencing or high density SNP chips, also the actual phenotypic records. And most important thing is bigger the training set, that is the animals or plants with both genomic and phenotype data, larger the training set, better is the accuracy of prediction, estimating genetic breeding values and ranking them. So the data size does matter, but as I've said, this is not something on the level or on par with some of the deep learning algorithms and machine learning that requires a massive amount of data on training to try and come up with a better prediction accuracy. We can deal also with small to medium-sized data sets, as small as you've seen, around 2,800 animals in the dairy cow. So we are able to lift the accuracy and reliability even with a smaller data set, but bigger the better. And I would say—
all right, thank you, Haja. I think we're 5 minutes over roughly. If there's a last burning one, we'll deal with that. If not, we'll draw it to a close, and I'll say thank you for attending. Please look out for the recording on social media, and then also for the next one in our series of webinars. And with that, I will wish you all a great summer, and thanks again for attending.
Thank you.
Bye-bye.
Bye, everyone. Bye.